Okay, let's unpack this. Have you ever popped a pill, trusting it'll make you feel better, but then stopped to wonder, what else is in here besides the medicine?
Yeah, it's a thought, isn't it?
It really highlights just how much trust we put in pharmaceuticals. But the truth is, making medicines is, well, incredibly complex, and sometimes unwanted guests try to sneak into our prescriptions.
And what's fascinating here is that these unwanted guests aren't just minor inconveniences. They can seriously impact the safety, efficacy, and overall quality.
Right.
Think of it this way. Every single dose you take, it's gone through rigorous checks to ensure its purity. It's not just about preventing obvious harm either.
Mm.
It's about making sure the drug works exactly as intended, preventing maybe subtle side effects, and critically, maintaining patient trust. For the industry, that trust is maybe their most vital asset.
That makes sense.
So today we're taking a deep dive into this hidden world of pharmaceutical impurities and the ingenious limit tests that act as their ultimate purity patrol.
Okay, good. We've been sifting through some crucial documents on pharmaceutical inorganic chemistry, specifically digging into the details around these lurking impurities and, the foundational tests used to catch them.
Right.
Our mission today, to give you a shortcut to understanding what these impurities are, where they're coming from, and how scientists make sure our medications are as pure as they absolutely need to be.
Think of it as a behind-the-scenes peek at quality control.
Exactly.
Yeah.
Helping you appreciate the meticulous science that goes into every single dose you take.
Hmm.
So first things first, what exactly are these impurities? Are we talking like a little bit of dust or something potentially more impactful?
Well, impurities in pharmaceutical substances are essentially any unwanted chemicals or contaminants. They can pop up in the drug substance itself, the API, active pharmaceutical ingredient.
Right, the main stuff.
Exactly. Or in the excipients, those, inactive ingredients like fillers, binders,
Okay.
or even in the final product you hold in your hand, they're basically anything that's not the intended pure compound.
And they can mess things up.
Oh yeah, they can compromise everything from how well the drug works to, well, patient safety. Big time.
Okay, that makes sense. So if they're not supposed to be there, where do these unwanted chemicals even come from? It's not like they just poof, magically appear.
Exactly. No magic here. Their sources are incredibly diverse. They span the entire life cycle of a drug. Seriously,
The whole thing.
the whole thing. We're talking everything from the very beginning, raw material procurement, all the way to how the final product is stored on the shelf. It's like a scientific detective story trying to track them down.
Wow.
Let's break down some key origins, maybe.
Yeah, let's do it.
Okay, first, you got to consider the raw materials. The active pharmaceutical ingredients themselves, the APIs. They can come with leftover solvents from making them, or byproducts from the synthesis reaction, or even stuff that forms if they sit too long, degradation products.
Okay.
Even the excipients, those so-called inactive ingredients, they can carry impurities from their own raw material sources or how they were made.
Huh. Never thought about the fillers having issues.
Right. And sometimes it's physical contaminants. Things like microbial contamination, heavy metals are a big one, or even just foreign particles like, believe it or not, dirt, oil, or even hair.
Hair. Seriously.
Yes, hair. If controls aren't tight, then there's the manufacturing process itself. Reagents and solvents used in making the drug might leave residues if they're not cleaned out properly.
Leftovers.
Exactly. Leftovers. You can also find unreacted bits from earlier chemical steps, intermediate products. And then there's a huge one, cross-contamination.
Ah, like if they make different drugs nearby.
Precisely. Imagine different drugs being made in the same facility.
Yeah.
Impurities from one batch literally jumping over to another if procedures aren't perfect.
Yikes.
Then we also have degradation of active ingredients. This can be chemical degradation, exposure to heat, light, humidity, oxygen, and work. It can break down the API, forming new impurities.
So light can actually change the medicine.
Absolutely. Or it can be microbial degradation. Microorganisms can actually eat or break down APIs and excipients during manufacturing or storage. That's particularly worrying.
Yeah, definitely.
Packaging materials are another maybe surprising source.
The bottle itself.
Yeah, or the blister pack.
Yeah.
Chemicals like plasticizers or stabilizers can leach out of plastic, rubber, or glass packaging into the drug. We call these leachables and extractables.
Huh.
Plus, just handling during packaging can introduce dust, oils, residues from human contact or the machinery.
Okay, wow.
And of course, storage conditions, they play a huge role. Environmental factors like temperature swings, humidity, light, oxygen, all can cause chemical breakdown or let microbes grow, even just leaving a container unsealed.
Right, makes sense.
And we absolutely have to talk about microbial contamination. Bacteria, things like Pseudomonas aeruginosa or Staphylococcus aureus fungi, yeasts, even endotoxins from bacterial cell walls. They can get in during manufacturing. This is super critical for injectables and liquids.
Yeah, you don't want that injected.
Definitely not. Finally, even human handling and environmental contamination are significant. Operators can introduce impurities, oils, dirt, chemical residues just through contact if good manufacturing practices, GMP, aren't followed strictly.
GMP. Okay.
And airborne particles, dust from the production environment itself, can just settle in. It really is an all-encompassing challenge.
That is an incredible list. It really makes you realize just how many points of vulnerability there are. So with such a diverse rogues gallery of potential intruders, how do scientists even start to make sense of them all? How do they classify these hidden nasties?
Yeah, it's a lot. They typically fall into a few major categories to manage it all. First, organic impurities. These are chemical impurities directly related to the API or the excipients, often from how they were made or if they broke down.
Like those leftovers you mentioned.
Exactly. Unreacted starting materials, byproducts, residual solvents. Then there are inorganic impurities. These include trace metals, lead, arsenic, mercury, things like that.
The really bad stuff.
The really bad stuff. Highly regulated because they're extremely toxic, even in tiny, tiny amounts. This group also includes salts or other elements like iron or zinc, often coming from raw materials, equipment, or just the environment.
Okay.
We also classify microbial impurities, bacteria, fungi, endotoxins. Again, critical for liquids and injectables.
Right.
Then physical impurities. These are actual foreign particles, not chemically part of the drug. Think tiny glass fragments, metal bits from equipment wear, plastic particles from containers, dust, fibers.
Actual physical bits.
Actual bits. And finally, adventitious contaminants. These are sort of unintended incidental impurities from external sources. Maybe leftover cleaning agents, lubricants from machinery, or that cross-contamination we talked about from other products made nearby.
Okay, so we've identified our unwanted guests, and it sounds like a truly formidable lineup, but knowing the enemy is only half the battle, right?
Right.
Our purity patrol needs some serious tools. How do they actually prove a drug is pure enough? Is that where these limit tests step in?
Indeed. Limit tests are really the frontline defense in pharmaceutical purity. They're specific tests designed to identify and, importantly, control small quantities of specific impurities in a substance.
Small quantities, okay.
Their main purpose is to ensure that a product meets the really stringent purity standards set by official pharmacopoeias.
Pharmacopoeias, those are like the official rule books.
Exactly. Official national or regional books of standards, like the Indian Pharmacopoeia, the IP, or the United States Pharmacopoeia, the USP. They set the bar for drug quality globally.
Got it. So it's not just a simple yes, no, is it there or not? It's more like how much of it is there.
Precisely.
And are these tests mostly for the inorganic stuff you mentioned, like the metals?
Generally, yes.
Mm-hmm.
While analytical chemistry has tests for everything, these specific limit tests we're focusing on are typically carried out to determine those inorganic impurities. And the core principle is quite clever, actually.
Oh, yeah. How so?
Well, the impurity you're looking for reacts with a specific chemical, a reagent, and this reaction produces either a visible color or a precipitate, which basically makes the liquid cloudy or turbid.
Ah, so you can see it.
You can see it. This visual change, how intense the color is or how cloudy the liquid gets, is then compared to a standard solution.
A standard?
Yeah, a solution that contains a known, accepted amount of that same impurity.
Mm.
If your test sample shows less turbidity or color than the standard, it passes.
And if it's more cloudy or colorful?
It fails.
Yeah.
Simple as that. It's a clear visual quality check built on chemical reactions.
Okay, this raises an important question. Given all these potential impurities, the sources, the complexity, why are these limit tests so incredibly vital? What are the real stakes here?
Oh, their importance truly can't be overstated.
Mm.
Yeah, you really can't stress enough how important these are. This level of meticulousness ensures that every dose you take isn't just safe, but also fully effective. It's the difference between a medication that genuinely helps you and one that could, well, introduce new problems or maybe just not work as well as it should.
Right, efficacy.
Exactly.
Yeah.
When we talk about patient safety, the very foundation of public health, these tests are absolutely non-negotiable. They are like the silent guardians of your health, working behind the scenes.
Silent guardians. I like that. Making sure what you're putting into your body is precisely what it claims to be.
That's the goal.
Okay, let's get into the nitty-gritty then. How do they actually test for specific impurities, like, say, chloride or arsenic? Each one must have its own special approach, right? Like a unique detective tool for each contaminant.
Absolutely, you nailed it. Each impurity needs a tailored approach. They leverage different chemical reactions to make that specific impurity visible. It really is like having a specialized set of detectors for different types of uh, ghosts in this system, so to speak.
Ghosts in the machine
Yeah.
or medicine. Okay, tell me about chloride first.
Okay, for the limit test for chloride, we use its reaction with silver nitrate. Simple chemistry, really. When chloride ions meet silver nitrate in the right acidic environment, using nitric acid, they form silver chloride.
And that's the cloudy stuff.
That's the cloudy stuff, AgCl. It shows up as turbidity or cloudiness. The more chloride you have, the cloudier it gets. So you prepare your sample solution, and you compare its cloudiness side by side with a standard solution containing the maximum acceptable amount of chloride.
In special tubes. Right. Nestler cylinders.
Yep, usually in Nestler cylinders for easy comparison. If your sample is less cloudy than the standard after, say, 5 minutes, you're good to go. It passes.
Okay.
Oh, and a crucial detail here. You have to use distilled water for these tests.
Why is that?
Because regular tap water usually have chloride ions in it already. It would totally interfere and give you a false positive, making you think there's more impurity than there is.
Ah, good point. Okay, what about sulfate? Similar idea.
Very similar idea. For sulfate, we use a reaction with a barium sulfate reagent, usually in the presence of dilute hydrochloric acid. This forms a barium sulfate precipitate, BaSO4, which again shows up as turbidity.
Barium sulfate. Isn't that what they use for medical imaging?
The very same, yeah.
Yeah.
But here, we're just forming a tiny bit to see the cloudiness. It's the same visual comparison game. Less cloudy than the standard means it passes. Same setup, usually Nestler cylinders, wait 5 minutes, compare.
Okay, chloride, sulfate, turbidity tests. What about iron? You said that one's different.
Right, the limit test for iron is a bit more like a magic trick, color change magic. Any iron impurities present, usually as ferric ions, Fe+3, are first chemically changed, reduced to ferrous ions, Fe+2.
Okay.
Then, in an alkaline environment, which we create by adding ammonia solution, these ferrous ions react with something called thioglycolic acid to form a vivid reddish-purple colored complex. Ferrous glycol.
Reddish purple. Got it.
And the intensity of that color tells you how much iron is present. More iron, deeper color.
You mentioned adding citric acid, too. What does that do?
Ah, good catch. The citric acid is clever. It forms a soluble complex with the iron first. This prevents the iron from crashing out of solution as iron hydroxide when we add the ammonia to make it alkaline. It keeps the iron available to react and form that telltale purple color.
Smart. And you have to compare it quickly.
Yes, that ferrous thioglycolate color is sensitive to oxygen in the air. It fades pretty quickly due to oxidation. So you have to make the comparison against the standard almost immediately. It's a bit of a race against time.
Yeah, okay, interesting. Now this next one, the Gutzeit test for arsenic, I hear it's quite unique. You said they don't even use a Nestler cylinder. What's up with that?
That's right. This is where things get really unique. Arsenic impurities, when you treat them with certain reagents like zinc and acid, they get reduced and form arsine gas, ASH3.
A gas.
A gas. So if you used a standard Nestler cylinder, the arsine would just poof, escape into the air, and you wouldn't detect anything.
Right, okay.
So the Gutzeit apparatus is specifically designed to trap this gas. It's basically a sealed reaction vessel with an outlet tube.
Like a ghost trap for arsenic gas.
Kind of. The escaping arsine gas is directed through this tube, where it passes over a special paper strip impregnated with mercuric chloride, HgCl2.
Mercuric chloride paper.
Yep. The arsine gas reacts with the mercuric chloride, and it forms a distinctive yellow stain on the paper. The intensity of that yellow stain tells you how much arsenic was present. You compare the stain on your test paper to a standard stain prepared at the same time.
Clever. And what about that lead acetate wool you mentioned?
Ah, another key detail. During the reaction, other gases might also be produced, like hydrogen sulfide, which could also react with the mercuric chloride paper and interfere, giving you a false reading.
Okay.
So you place a little plug of cotton wool soaked in lead acetate solution in the tube before the mercuric chloride paper. The lead acetate reacts with and traps interfering gases like hydrogen sulfide, ensuring only the arsine reaches the paper to produce that accurate yellow warning stain. Very neat. Okay, lead. That sounds particularly nasty to have in medicine. Is it a common impurity? How do they tackle that one?
Yeah, lead is considered one of the most undesirable impurities, really, because it's highly toxic, even at very low levels. It can creep in from various sources. Maybe a sulfuric acid used in some chemical processes contained lead, or lead-lined equipment was used historically, or even from the glass bottles chemicals were stored in.
Wow, even the bottles.
Even the bottles. So testing for it is crucial. For lead, they use another clever color change test. It involves a reagent called diphenylthiocarbazone, which most people just call dithazone.
Dithazone, okay.
Dithazone itself, dissolved in something like chloroform, is green. But when it reacts with lead in an alkaline solution, it forms a lead dithazenic complex, which is strikingly violet. This mixes with the green, resulting in an overall red color appearing in the chloroform layer.
Red for danger, basically.
Pretty much. The intensity of the red color is proportional to the amount of lead. This test is usually done in a separating funnel, those pear-shaped glass funnels with a stopcock at the bottom. Yeah, I know them. Why there?
Because it involves extraction steps. You shake the aqueous solution containing the lead with the dithazone solution in chloroform. The red lead complex gets extracted into the chloroform layer, separating it from other stuff. You also add other reagents like ammonium citrate, potassium cyanide, carefully handled, of course, and hydroxylamine hydrochloride. These help adjust the pH and mask other metal ions that might interfere with the dithazone reaction, making the test specific for lead.
Sounds complex but precise.
Very precise. It has to be for something like lead. Then you compare the final red color visually against a standard.
Okay, what about just heavy metals in general? Is there a catch-all test?
There is, yeah. The limit test for heavy metals. This is a more general test designed to detect impurities of various heavy metal, things like cobalt, tin, manganese, copper, silver, antimony, and including lead as well.
So it's like an umbrella test.
Exactly.
Yeah.
It's based on the reaction of these metallic impurities with either hydrogen sulfide gas, H2S, or sodium sulfide solution, Na2S, in an acidic medium. This reaction produces metal sulfides. And what do they look like?
They typically appear as a brownish or blackish color or precipitate. The intensity of this brown color is compared against a standard lead solution treated the same way.
So lead is the benchmark even here?
Yes, the impurity level is usually expressed in terms of lead parts per million, or ppm, even if other metals are causing the color. To give you a sense of the stringency, a common limit might be something like not more than 20 ppm. If a sample produces more color than the 20 ppm lead standard, it fails.
20 parts per million? That sounds incredibly tiny.
It is incredibly tiny. We're talking trace amounts. And there are actually different methods for this test. Methods A, B, C, D, depending on whether you use H2S gas or sodium sulfide solution, and whether the substance you're testing dissolves clearly under specific conditions. It adapts the test to the sample.
Okay, that covers a lot of ground.
Yeah.
But you also mentioned modified limit tests earlier for chloride and sulfate. Why would a test, after all this precision we've discussed, need to be modified? That's a great question. Sometimes the standard limit tests just don't work effectively. The main reason: colored compounds.
Ah, right. If the sample itself has color.
Exactly. How can you reliably detect a faint turbidity, cloudiness, or a subtle color change if your starting solution is already, say, yellow or pink or brown?
You wouldn't be able to see it properly.
Precisely. It would be like looking for a gray ghost in a smoky room, as you said.
Yeah.
You just can't make an accurate visual comparison against the standard. That's when we need a modified approach to basically clean up the sample first.
So how does the modified chloride test work?
For the modified limit test for chloride, used for these colored compounds, the key modification is adding a decolorization step before you do the normal chloride test procedure.
Decolorization. How?
Typically, the sample is treated with potassium permanganate, KMnO4, which is a strong oxidizing agent, often with a little ethanol, and then heated. The permanganate oxidizes the color organic impurities. Then you filter the solution to remove any manganese dioxide formed.
And that leaves it clear.
Ideally, yes, or at least much less colored. This clear or decolorized filtrate then goes through the standard chloride test. Add nitric acid, add silver nitrate, wait 5 minutes, compare the turbidity against the standard in Nessler cylinders. The core principle of silver chloride formation causing turbidity remains exactly the same. We just remove the interfering color first.
Makes sense. And the modified sulfate test, similar idea.
Very similar concept for the modified limit test for sulfate. It's also designed for colored compounds where the normal sulfate test wouldn't give a reliable visual comparison.
So a decolorization step here too.
Yes, the procedure usually involves a similar decolorization step, often using potassium permanganate and maybe ethanol, followed by filtration, just like for the modified chloride test. After decolorizing, you proceed with the sulfate test.
Which involves barium chloride, right?
Right. The core principle of forming barium sulfate, precipitate from soluble sulfate ions reacting with barium chloride,
Mm.
usually in the presence of acetic acid this time, remains the same. But the modified test might also include specific additions, like sulfate-free alcohol or a tiny bit of potassium sulfate.
Why add those?
The alcohol helps prevent something called supersaturation, where the barium sulfate might not precipitate out properly, and it ensures any turbidity that does form is fine and uniform, making it easier to compare. Adding a seed crystal of potassium sulfate can also help initiate precipitation consistently. It's all about increasing the test's sensitivity and reliability for these challenging colored samples.
Wow. The level of detail and thought that goes into just making sure these tests work correctly is amazing.
It really is. It has to be.
Phew. Okay. That was a truly deep dive into the hidden world of pharmaceutical impurities and this incredible purity patrol of limit tests. From, well, microscopic dust and leftover chemicals to heavy metals like lead and arsenic, it's crystal clear that ensuring medicine quality is absolutely no small feat.
Not at all.
It really makes you appreciate the unseen science protecting us every day.
And if you connect this back to the bigger picture,
Mm.
all that precision we talked about, the need for specific reagents for different impurities, the specialized equipment like the Gutzeit apparatus, the modifications for tricky samples,
Yeah.
it all just underscores the immense responsibility that pharmacists, chemists, manufacturers, everyone involved carries.
Yeah.
It's a huge testament to the rigorous science that protects us, ensuring
Yeah.
the medications we rely on are truly what they claim to be day in and day out.
So what does this all mean for you listening? Well, next time you reach for that cough syrup or maybe a pain reliever or any medication, really, maybe take just a second.
Mm-hmm.
Take a moment to appreciate the invisible guardians, those dedicated scientists, and these meticulous limit tests that worked tirelessly behind the scenes. They're ensuring that what you're putting into your body is not just effective, but also as pure and safe as humanly possible.
Absolutely. It's a hidden layer of safety we often take for granted.
It really makes you think about how many layers of unseen science are constantly working to protect us, doesn't it?
It definitely does.
Something to mull over, perhaps, until our next deep dive.